Bring Skywalker to me. Only joking, it's me. And here is a video with your workbook and examples for natural selection. So here we go. Things that you'll need. And you might have your textbook handy, but certainly your workbook. I'm not going to give you the answers here, I'll just give you some teasers and the theory you need for the examples. Hopefully you're experts in natural selection now, and we chatted through the finches example last time. And the key message from this is that any population will have variation in the individuals there. These will have differential reproduction. So some of them will survive and reproduce, some of them won't. And those that do are the ones that have this selective advantage. They're fitter and they'll pass on the favourable alleles to the next generation. And that will affect the allele frequencies in the gene pool over time. So the favourable alleles will increase. That's just a quick recap of the theory. Let's have a look at your workbook starting at page 10. Eventually you'll get the tutor version, which you'll be able to mark yourself, or maybe your teacher will look at that for you. On page 10, just a load of bump really, recapping the theory that we've done. And page 11, there's at the top a little graph there. That's just showing over time, when we said about the allele frequencies. You've done this in Hardy-Weinberg anyway, but the allele frequencies for the favourable alleles will increase, and the ones that are not favoured go down. Summarise the sequence explanation for natural selection. It's just where we started with this PowerPoint and in your theory, so you can do those. Those are fine. Fill in the box of explanation. Mutation, we did a bit on that in the first PowerPoint. Sexual selection, the fittest individuals survive and reproduce. Gene flow. And that's always a bit of a tricky one. Gene flow. It's just really the changes in the genes from generation to generation. Genetic drift, particularly important in small populations where there's genetic changes due to mutations and so on. So it's really the change in the alleles present. And natural selection, the mechanics of all of this. So find out those definitions, and more bump and something to look at. But here's something that you might not know that much about yet: density dependent and density independent factors. These are factors that affect populations. These could be selection pressures, but the dependent factors depend on how big the population is. So an easy way to think of this is for food. You need food to survive and then reproduce. But if there's a limited amount of food, then the more population you have, then the harder it is to find food. So an increase in the population makes food an increasingly scarce resource. So any factor that depends upon the size of the population. So other things like competition for nesting places, light. If you're a plant, the more plants, you get less light due to shading and things like that. Density independent is a factor that will affect all the population equally. It doesn't depend on how many are there. So if you imagine a forest fire that will kill all of the population of something, it doesn't matter whether there's 5 50, 500 of whatever the animal is. If there's a big forest fire, it'll kill all of them. And also things like flooding. Flooding will affect. It might kill off a certain type of plant. It doesn't matter how many plants are there. They're all going to get killed. So it's not dependent on the population level. And more blurb on page 13 and 14. Then there's a table to fill in on page 19 and 20. Examples of natural selection. So we'll give you a bit of help with this. Your table has got 1, 2, 3, 4, 5 things down the left. So down the left, these are just the points about natural selection that you know about by now. And we've given you one example, which is stabilizing selection, sickle cell trait, and malaria. So that would be the first one. I'll chat through that, and then we've got a rats example with warfarin and bacteria example with penicillin. So let's chat about those. Here's a mosquito. Mosquitoes carry a parasite, a single-celled parasite, and when the mosquitoes land on you, they pierce your skin, and the mosquitoes feed on your blood, and that's how they get their nutrition. But they do infect you with this parasite, this single-cell parasite, which can then live in your red blood cells. And this is a problem, as it's very hard to get rid of this Plasmodium parasite, because with it being living in your cells, a lot of the drugs that we have are ineffective. It's not a lot of antibiotics, things like against bacteria won't work, because this is a eukaryotic parasite. So very difficult to treat. So there's our red blood cells, and you are having two alleles of a particular gene. The gene that we're going to have here is HB, standing for hemoglobin, and normal people are homozygous for A. So your hemoglobin is normal hemoglobin, and you know a lot about hemoglobin from your first year work. We'll talk about malaria in a moment. People who are heterozygous for the HP gene with alleles A and S. Well, this allele S, standing for sickle, this affects the hemoglobin and makes it kind of more fibrous instead of globular, and that affects the shape of the red blood cells. And as you can imagine, if you've got a sickle shape red blood cell, that's not as good at carrying oxygen as the normal hemoglobin and the normal shaped red blood cell. And if you are homozygous for the sickle, so HBS, then all of your red blood cells are sickle shaped. And this is really quite catastrophic for your health and carrying oxygen around your body. So normal people, homozygous for A, heterozygous people, and homozygous for S. So if there was no malaria present, you can imagine, so in this country, in the UK, most people are going to be homozygous for A. If you were heterozygous with a sickle, then you might imagine you'd be slightly affected by the sickle cell, but generally health is okay, and that's called sickle cell trait. And these people here, homozygous for S, they're very ill individuals, so they're not selected for. So in the UK, the best is going to be homozygous for A. The thing about malaria is that the malarial parasite will live quite happily in normal red blood cells, and it'll thrive in normal red blood cells. But anything that's got this altered hemoglobin, the malarial parasite cannot survive in this type of blood very well, and also it can't survive in full sickle cell. So in parts of the world with malaria, sub-Saharan Africa, for example, then individuals that are heterozygous, these are protected against malaria to a large degree, and these are the individuals that are selected for. So normal individuals are susceptible to malaria. Sickle individuals are very ill from sickle cell disease, but the heterozygotes are in the best position there. So in these places with malaria, there's a lot of people that are in this position, and those will, if you think about it from natural selection, those will survive, those will reproduce, and they'll pass on these alleles to the next generation. So there'll be A's and S's in the population. This will, of course, you've done genetics, this will lead to normal people and sickle people being produced, but we'll get A and S in the population. So you just need to put that story into your natural selection theory, and we've done that one for you. Another example, genetics of warfarin resistance in brown rats. It's a little bit like the previous example. So we've got normal rats. These are the best favoured if there's no rat poison. So if people don't put down this warfarin rat poison, then the normal rats, SS, are favoured. There's the heterozygous rats, and they've got an allele which confers resistance to warfarin. But there is a side effect of this, and they need slightly better nutrition from their food. So they need this extra requirement for a vitamin K. And then we've got our third rats, which are homozygous, and they have this warfarin resistance, homozygosity. But as you can see, this requirement for vitamin K now goes up by 20 times. So they've got this massive requirement for this vitamin, and they're unlikely to get that through their normal diet. So two situations: if there's no warfarin about, no rat poison, then the rats that are fittest, most likely to survive and reproduce, are the normal rats, as they will outcompete either of the other two rats that need vitamin K. If there's warfarin about, then the normal rats will get killed by warfarin. The double RR, so the homozygous rats, they'll be resistant to warfarin, but because they need all of this vitamin K, massive requirement, they will probably not survive either. So it's the heterozygous rats that will survive, reproduce, pass on these advantageous alleles, both S and R, to the next generation. And the allele frequencies for S and R will both remain high in the population. So that's rats. And the last one, antibiotic resistance. You've done this at in your AS anyway. We've got our normal bacterial population, and if you treat these with antibiotics, antibiotics will kill these bacteria. So, for example, penicillin. You get mutations, so change in the DNA, and that might make these one of these bacteria resistant to the antibiotic. So when you treat this population containing both some resistant bacteria and the normal population dies, and the population with the resistant allele will grow, reproduce. The interesting thing is that when you treat, and this is a problem with superbugs in hospitals, if you get rid of all of the non-resistant bacteria, the resistance bacteria have very little competition for food and space, and so they can quickly overcome and build up a large population. So that's the problem with things like MSRA or C. difficile in hospitals. So again, you've just got to tailor your natural selection story to mutations. This is your variation. You've got your selection pressure from the antibiotic. Some individuals have got a selective advantage. They are fitter. They will survive, reproduce, pass on the advantageous alleles to the next generation, and the allele frequency will increase for those alleles. So do your workbook and mark it or get it marked.